Hey guys, so today our group is going to talk about CRISPRainbow. I'm Paul.
And I'm Amanda.
So you might have heard of a technique established over 10 years ago called Brainbow. What Brainbow basically is that it's a technique for labeling the mice neurons with fluorescent proteins. What we do over here is that we insert the gene which encodes for the fluorescent proteins into the mice. The mice then produce the fluorescent proteins in vivo. Initially, this technique shows great promise because it is able to produce a highly detailed image of the mice neuron. However, the fact that we have to select for transgenic cells and then further breed the cell lines means that this technique is very time-consuming and not so efficient. 9 years later, a technique called CRISPRainbow was established. CRISPRainbow further builds on the principles established earlier by Brainbow. It has similar working principles in that it uses fluorescent proteins to label the cell. However, it's different from Brainbow in that it doesn't involve the insertion of a fluorescent protein encoding DNA, thus making it more efficient. CRISPRainbow also involves the use of the CRISPR and Cas9 system. So what is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is a system of adaptive immunity in bacteria. So as you can see in this diagram, there's a bacteria and there's a virus. Say a virus called a bacteriophage injects its DNA into the bacteria. The bacteria then has a Cas nuclease which cuts the bacteria up and inserts the DNA into the CRISPR locus. The CRISPR locus then encodes for a CRISPR RNA, which is further processed into smaller RNA called guide RNAs. These guide RNAs then bind with the Cas9 nuclease, creating a guide RNA Cas9 nuclease complex. When the bacteria is infected by the same type of virus again, this guide RNA Cas9 nuclease complex recognizes the foreign DNA and cuts it up. Thus, the bacteria survives. So, what has CRISPR-Cas9 system been used in research? The CRISPR-Cas9 system has been used in research, particularly in the molecular biology field. Researchers have recognized the potential that they can engineer a guide RNA which binds to the DNA and recruiting the Cas9 nuclease to a specific site. The Cas9 nuclease, once bounded, will introduce a double-stranded break to the DNA. There is also another use for CRISPR-Cas9 system, which is in genome labeling. So, how do you label with CRISPR? Labeling with CRISPR requires another type of Cas9 called the dCas9, or the nuclease-dead Cas9 protein. This type of Cas9 protein has a similar function to the normal Cas9 protein, but it is different in that it has the nuclease function removed, which means that when it is bounded to the DNA, it will not introduce a double-stranded break. The problem with only having one type of dCas9 is that we will not be able to distinguish between different types of guide RNAs. So researchers solved this by using orthogonal dCas9. It has similar function to the normal dCas9, but it will bind to the specific guide RNAs. So researchers will obtain the orthogonal dCas9 from these three bacteria.
Alright, so now that we can label different sites on the DNA, we want to label them with different colors. And this is done by recruiting fluorescent proteins to the guide RNA. So as you can see, this is the structure of the guide RNA. It consists of this complementary region, which is complementary to the site that you want to target. And the rest of the guide RNA consists of a series of stem loops. So these stem loops can be used to recruit RNA-binding fluorescent proteins. So in CRISPRainbow, they started off with these three guide RNAs. So as you can see, the first guide RNA, it has two stem loops, both of which recruit the blue fluorescent protein. So any sites that are labeled with this guide RNA will fluoresce blue, as you can see here. Similarly, the other guide RNAs, they have one that recruits green fluorescent proteins and another that recruits red fluorescent proteins. So the question is, we have the primary colors red, blue, and green. How do we make more colors? Well, we can use combinations of different fluorescent proteins to make different colors. So it's the same principle as the pixels on your phone or your computer. Every color that you see on a screen is actually just a unique combination of different intensities of red, blue, and green light. Similarly, on the molecular scale, we can do this with fluorescent proteins. If you have different combinations of blue proteins, fluorescent red proteins, and green proteins, you can make different colors. For example, if you have blue proteins and green proteins in the same vicinity, what you see on the fluorescent microscope is cyan. And this is exactly what CRISPR-Rainbow does. So let's take this guide RNA, for example. The first stem loop recruits the blue fluorescent protein, and the second stem loop recruits the green fluorescent protein. So any sites that are labeled with this guide RNA will have both blue and green fluorescence, and when you merge them, you actually see the color cyan. They also created guide RNAs that would produce magenta fluorescence, yellow fluorescence, and even white fluorescence by combining all three of the fluorescent proteins. So because each of the guide RNAs can be engineered to target a different site in the genome, using the 6 RNAs, not including the white one, we can label up to 6 sites in the genome. And this is what the CRISPR-Rainbow paper did. As you can see, they were able to label up to 6 sites uniquely with different colors, and these all represent different sites in the genome. So what are the advantages of CRISPR-Rainbow? As we've mentioned, it's able to label multiple sites. It's highly specific because you can engineer the guide RNA to target only certain sites in the bacterial genome. And theoretically, you can produce any color just by using unique combinations of the fluorescent proteins. And I guess one of the most important advantages of this technique is that you can do real-time imaging. So typically, if you wanted to label a cell with many fluorescent dyes, you would have to fix the cell in formaldehyde and then dye it. But this actually kills the cell, and you won't be able to actually observe what's going on in a real cell. But if you use CRISPR-Rainbow, because it doesn't affect the workings of the cell, you're just putting in guide RNAs that bind to the genome in certain places, you can actually observe how the genome changes over time or in response to different stimulus. There are disadvantages. It's been suggested that the CRISPR-Cas9 system is inherently flawed because it can exhibit off-site binding up to quadruple mismatches. What this means is that when the Cas9 and RNA complex attempt to bind to the genome, the binding may not be completely complementary, but it will still bind to that site. There's also difficulty in delivering the Cas9 plasmid into the cell in the first place to express the system. Despite the advantages, it has been proven effective to the point that it's actually commercially available for $375 US. What are the uses of this system? So we've said that it's a system for genomic labeling, and though it sounds simple, it actually has a lot of applications. For example, you can measure the distance between loci. So if you label 2 genes on the same chromosome, and you know the genomic distance between these, you can and using CRISPR-Rainbow, you can find the physical distance between the genes, and it becomes a link between physical mapping and genome mapping. If you label loci on different chromosomes, you can identify where different chromosomes are, and then you can study DNA compaction. Lastly, as I've mentioned, because it is a real-time monitoring, you can also study dynamic processes like DNA replication, RNA damage repair, and DNA rearrangement. This is one example of the uses that CRISPR-Rainbow has been put to recently. This is from earlier this year. And in this paper, CRISPR-Rainbow was used to actually track the movement of telomeres in cells in real time. So in conclusion, CRISPR-Rainbow is a system using nuclease-dead Cas9 and guide RNAs to label multiple genomic sites with different colors. It's a novel technique, and it's also been suggested to be very, very promising, and I'm sure we'll see many more papers on this in the future. Thank you. These are references.